Okay, we're going to start talking about chemical equations so that you can understand how chemical equations work. Basically, chemical equations mean on one side there are reactants, things that react together. And remember, to be a reactant, it has to have an empty spot in its valence shell. And when you put those two things together, you get a product. Like if you take sodium atoms and chloride atoms, add them together, they will bond ionically and become salt, table salt. Here is a chemical reaction that is called the bicarbonate buffer system. This is an example of a chemical reaction that occurs in the blood. This keeps your blood pH at the right level. If your blood gets too acidic or too alkaline, it can have very serious effects, even kill you. So on one side, we have carbonic acid, H2CO3. On the other side, we have bicarbonate ion plus a hydrogen ion. Okay, the bicarbonate's a negative ion, a hydrogen ion is a positive ion. A carbonic acid is not charged. So, if your blood gets too alkaline and it needs to increase the acidity, the carbonic acid here will be broken up into separate bicarbonate ions and hydrogen ions. What determines the acidity of a solution is a hydrogen ion. Write that down. The more hydrogen ions you've got, the more acidic the solution. Okay? Now, if the blood gets too acidic, these extra hydrogen ions can be bound to a bicarbonate ion to make this carbonic acid. Carbonic acid is a very weak acid. A hydrogen ion is a very strong acid. Okay? That leads us into a brief talk about pH scale. pH scale ranges from 1 to 14. Okay? With 14 being the most alkaline or basic, those are synonyms, and 0 being the most acidic. Okay, very strong acids. On here, we can see several examples of various items and how acidic they are. Coca-Cola is very acidic. Tomatoes are pretty acidic. Milk is a little acidic. 7 is true neutral. 7 means it is neither acidic nor basic or alkaline. It's just neutral. But you go above 7, like to 8, that's going to be 10 times more alkaline than 7. 8 and 9. 9 is 10 times more alkaline than 8. Now this is where it gets tricky. This is a scale, and each step on the scale is a multiple of 10. So 7 is neutral. 9 is 100 times more alkaline than 7 because it's 10 times here to 8 and then another 10 times here to 9. If we went between 7 and 10, that would be 1,000 times more alkaline. Let's say we compare 6 to 8. It's 2 steps. 10 times, 100 times. All right? And so it's a multiple of 10 between every scale point as far as how acidic or alkaline it is. Okay? Organic molecules. An organic molecule contains carbon, hydrogen. They have to have carbon to be an organic molecule. All right? Methane is a very common organic molecule. The primary organic atoms that make up organisms are carbon, hydrogen, oxygen, and nitrogen. You can use the acronym Ch2 to remember that. Those are the 4 that are the most important. All organic molecules will have carbon in them. Okay? There are many types of organic molecules. They're categorized into 4 units: carbohydrates, proteins, nucleic acids, and lipids. Lipids are also known as fats. Carbohydrates, proteins, and fats are very common in our diet. One of the reasons, we have a rather overweight society, and that's because we have so much food readily available. And the food that we have readily available is a lot of them are carbohydrates and fats. All right. And these are even affected by government policy as to what's the cheapest thing to eat. Government subsidies go to farmers who grow corn and wheat and things like that, these big cash crops, and they keep the cost of the wheat down and the corn down to consumers. Okay, it makes them cheaper because they're paid they grow so much wheat and corn that they're actually paid not to grow it so that it won't depress the price too low. They try to prop up the price that way, and it helps them keep certain foods very inexpensive. Unfortunately, the foods that are most inexpensive tend to be the ones that are the least favorable for you. If there were not government subsidies propping up these products, what would happen at first? The corn and wheat markets would become so inundated with corn and wheat that the price would drop beyond the ability for farmers to even survive. And so then most would go out of business, and the ones that would remain, that would bring the price back up. And instead of paying $4 or $5 for a box of cereal, you'd end up paying $20 or $30 for a box of cereal. Okay, so that's what it would really cost to produce them. And it still costs that much to produce corn and wheat, but they get money from the government to help supplement the costs. Okay, so they are able to sell wheat and corn at a loss. Okay, so maybe they sell it for $10 a bushel. It costs $40 a bushel to produce. So they would normally lose $30 a bushel. But government subsidies, state and federal, pay these farmers money. And that's where they make their profit. So they might get paid, get a subsidy of that equals out to $50 a bushel. So they can end up making a $10 a bushel profit. Monomers, organic molecules. A monomer is a single unit of a carbohydrate, protein, or nucleic acid. When they join together, they become polymers. So here is a protein polymer, and one of these structures, okay, is a monomer. Each one of these individual hexes for carbohydrates, and those are glucoses. Each glucose is a monomer, but when you put a chain of them together, you get a polymer. Okay, that's the difference between the two words. Now, how do we put two monomers together? It's called dehydration synthesis. Dehydration means take water away. Synthesis means put something together. So here are two monomers. Excuse me. And what you see is they'll bond. On this side of the monomer, they have an OH, and on this side, they have an H. So they will dehydrate this chain, pull this water molecule out, and in the process of pulling the water molecule out, they attach the two monomers. And now we have a polymer. Here is hydrolysis. Hydrolysis, hydro means water, lysis means to break apart. So if you take a molecule of water and add it to a polymer, it will take this polymer and split it into two separate monomers. So H2O, here's an OH and there's an H, and now we have two individual monomers. Those are two very important reactions. Monosaccharides are just monomers of carbohydrates. Glucose is the most important. Our bodies can only use glucose to generate energy. If we eat energy in any other form, like fructose, that's fruit sugar, our bodies have to convert this to glucose in order to burn it as fuel. Okay? Now there is two words here you need to know. There is a hexose sugar, 1, 2, 3, 4, 5, 6 sides, and then there are pentose sugars, 1, 2, 3, 4, 5. Okay? So here is dehydration synthesis happening. We're taking a glucose and a fructose. There's the H2O we're going to take out. And we make table sugar, sucrose. Some different carbohydrate molecules. There is starch. You find this in stuff like potatoes and carrots, things like that. Rice. We can digest starch. Glycogen is a storage substance of glucose molecules strung together. Okay, and this is our storage medium for glucose in animals. It's found primarily in places like the liver, things like that. Plants make starch. We can eat both of these. Cellulose, okay, these are all glucoses, but we can't digest them because this bond, we don't have any way of breaking that bond. To break that bond, you have to have bacteria that pre-digest it for you in some kind of a cavity. So cows and deer that eat grass have a rumen, R-U-M-E-N. A rumen is a cavity that has bacteria in it that will break the bonds on this cellulose chain so that we can then digest the glucoses, use them. A polysaccharide is just a type of polymer, but with saccharide, what we're doing is, instead of saying it's a polymer of carbohydrates, saccharides means carbohydrates. So we're just basically shortening the word. Instead of saying a polymer of carbohydrates, we just say a polysaccharide, and that means the same thing. Proteins. Proteins make certain structures. Okay, and their structures are far more variable. All of our enzymes are proteins. We have, there's egg protein, albumin, membrane channels on a plasma membrane, things that allow things to move back and forth between cells, going in and out of cells. These are muscle fibers, collagen fibers. These are for attachment and keeping things together, maintaining the integrity of certain things. And so there's all different kinds of proteins, okay? But they're all polymers. A protein, the monomers of a protein are called amino acids, all right? And all amino acids have some things in common. First off, amino acids are joined together to make polymers by a covalent bond called a peptide bond. You're going to want to remember that. Now you have a carboxyl group, you have an amino group. Every amino acid has this little NH2 and COOH, and then it'll have a little hydrogen down here. There's a carbon right smack in the middle, and then this R group is a variable group. This is what makes the difference between amino acids. There's lots of different amino acids. So the R group is very variable, and I can give you some examples. Here, the R group is just a hydrogen. That makes the amino acid glycine. If you put a sulfur, a hydrogen, and then this little CH2 up here, that's the R group, and it makes cysteine. Tryptophan, that great stuff you get on Thanksgiving Day when you're munching on your turkeys. It's got this very complex group of atoms stuck up here. That's its variable, or R group, and that's what makes it tryptophan. So, to put two amino acids together and into a polymer requires dehydration synthesis, and we can break them apart doing hydrolysis. But when we put the bond together here, when we put two of them together, that is called a peptide bond. Keep that in mind. Organic molecules. Proteins exist in a couple forms. Primarily, this is the primary form of a protein, if you take it and spread it out flat. Now, in nature, proteins either exist as twisted helixes or folded sheets. And so if you look here at this protein, that's its secondary structure. Its tertiary structure is the way it really looks in nature. Now, for an amino acid or a protein to function, it has to be in the right shape. If something happens to alter this shape, that protein will cease to function. Okay, and so if you look here, you'll see the flat areas, those are beta sheets, and then the little lines, those are twisted or alpha helixes. Now, sometimes you will take these proteins and you'll put them together. And you'll put, like here, there's 4 of them. This makes hemoglobin. That's called its quaternary structure. That's the 4th level. So primary is first, secondary is second, tertiary means third, quaternary means 4th. Okay. Nucleic acids. Nucleic acids include DNA and RNA. These molecules contain our genetic information. Here is a monomer of a nucleic acid. Every nucleic acid will have this phosphate group, and they'll have this, if it's DNA, it will be deoxyribose sugar. It's a pentose sugar, 5 sides. And then it has a nitrogenous base. There are 4 bases in DNA. All right? And these are the 4 bases of DNA: adenine, cysteine, guanine, and thymine. Okay? Now, RNA, instead of thymine, has uracil. So if you ever see a U, you know you're looking at RNA. If you see a T, you know you're looking at DNA. RNA has no thymines. DNA has no uracils. They are different versions of amino acids that are found in one or the other. Why do we have so few bases? Somebody asked me that once. Well, we really don't know how many bases the first DNA molecules had. Maybe they just had cytosine and guanine, and then they underwent modification and mutation, and we developed adenine and thymine, or adenine and uracil. That goes into a whole realm of the Miller-Urey experiments, which we will talk about later. Organic molecules, lipids. Another name for that is fats. Testosterone is a lipid. Okay. This is a triglyceride because it has 3 fatty chains. Okay. Okay. Let me see. What I want to talk about here. Saturated versus unsaturated fats. Saturated fats means that there are no double bonds. Now, each one of these little lines represents a bond between a hydrogen and a carbon. Saturated fats generally are found in animals, and at room temperature they're solid. Okay, like if you've ever taken the bacon grease, you don't pour it down your drain because it'll clog your drain. You pour it into a jar, and then it turns into kind of a solid, like a spread. Okay, and that's because every hydrogen and carbon are only connected by a single bond. Unsaturated fats have double bonds. Okay, so here's an unsaturated fat. Notice that between these 2 carbons, there are 2 lines. Each line represents a pair of shared electrons. So it's a double bond here between carbon and carbon. Now that means this is generally vegetable fats, and vegetable fats at room temperature are liquids. Okay, that's the only difference between unsaturated versus saturated fats. If you see commercials or things, they'll talk about, unsaturated fats are healthier because they're liquid. And I don't know if that's accurate or not. I've never actually researched that, but they're both fats, and there's so little difference. I don't see how there could be a huge difference, but again, I honestly don't know. I've never really researched that. Here is cholesterol. All right, now cholesterol gets high in it comes from fat, and it's made by your body. You need a certain amount of cholesterol. Now, some people genetically produce a lot of cholesterol, even if they have a perfectly healthy diet, they don't eat fats or very few fats, they can still have high cholesterol and have to take a drug for the cholesterol. Testosterone, okay? This is important in, obviously, healthy males. Low testosterone can lead to all kinds of things like suicidal thoughts, hyperaggression. I mean, there's just a lot of things. And for some reason, we are seeing dropping levels of testosterone in human males in this country. And we're trying to figure that out. There's your list of, if you know what all these words mean, you probably will understand what I was talking about in the lecture. So sometimes it's a good place to start. That wraps up chemistry. That's the bare minimum chemistry that you need. I mean, I've really stripped chemistry down to practically nothing. So hopefully that'll help you absorb this material. Thanks.